sa computer ko na. Excuse. Hello, attendees. We're going to start in 3 minutes. The time is 4:10. Good afternoon, everybody. I'm Apollo F. Olays IV of PAMIT USA Inc. Central California chapter. Before I move forward, I'd like to go over the house rules. Observe proper video conferencing etiquette. Use a computer or laptop preferably. Keep your microphone on mute unless asked by the speaker. Raise hand or post questions on the Q&A box after each session. Keep record of the PACE code. The PACE code will be announced before we end the session. PACE credits instructions available in the CCLC website post-registration tab. Lastly, be respectful. The last session of this convention is about point of care bioluminescent assay for the rapid detection of and diagnosis of UTI and AMR. Our guest speaker is an associate researcher at the University of Miami, Miller School of Medicine. He has a diverse education in microbiology, including medical technology, medical microbiology, molecular biology, and infectious disease. During his time at Far East University, he was involved with the technical committee and chair of the research committee for Nicanor Reyes Medical Foundation. He is also a national board exam reviewer for the Philippines. Ladies and gentlemen, let us welcome Dr. Sherwin Reyes. Take it away, Doc.
Hi guys. Do you hear me? Do you hear me clear?
Yes, we
Yes.
can hear you.
Okay, thank you. So, first of all, I would like to thank you guys for that warm introduction. So, I'm here to share with you my project for the discovery of a rapid diagnostic test for urinary tract infection, and later on we are verging to the rapid diagnosis side by side with antimicrobial resistance. Say, for example, is the patient suffering from UTI? Is the UTI or the organism um, that is infecting the patient has a capability of progressing to a resistant strain, or it has already the strain? So that is the coverage of the project that I am doing currently. And also I've been to many development of biosensors, since my laboratory in the Miller School of Medicine is focused on discovering of biosensor and a lot of other studies, especially of the microbiome and everything under the sun. So with that, let me share to you my screen. Do you see my screen, guys?
Yes.
Thank you. So for this afternoon, I will be sharing to you my research entitled "A Novel Cell Phone-Based Bioluminescent Assay for the Simple and Rapid Diagnosis of Urinary Tract Infection." So this is a simple way for us to diagnose urinary tract infection at a point of care setup. So before we proceed to the project, let me introduce to you diagnostic testing. So how are we going to do a diagnostic testing, and what are the criteria, or why we need to do diagnostic testing? So diagnostic testing is very essential, especially in the field of medicine, because for the clinician to have a good and final diagnosis of the case of the patient, we need to do certain testing, which include diagnostic testing. And this diagnostic testing should be, or should provide, a reliable and valid answer to clinical question, because this diagnostic testing is the one that will give uh, um, validation to whatever cause of an infection or the disease we are thinking of, especially those of the clinician. A sensitive and a specific laboratory test and imaging studies is very essential for accurate clinical diagnosis. A very poor clinical diagnosis may lead to patient suffering, and eventually can lead to death. Okay. Typically, the process involves a physical exam done by the clinician, evaluation of the symptoms, and later on, laboratory testing, wherein the medical technologies helps in the diagnosis of such diseases and infections. So where are tests performed? Clinical laboratory scientists or medical technologists do their testing in a clinical laboratory, wherein chemistry testing, microbiological testing is done in a secured, confined environment, wherein there is a lot of sophisticated materials or instrument needed for the diagnosis, plus the fact that these clinical technologies or clinical laboratory science has the knowledge to verify whatever the machine gives them as a result, because machine cannot think, but the clinical laboratory scientists can. Okay. And another test, we can do a point of care testing, wherein anywhere, maybe in a field, maybe bedside of the patient, or in the clinic of the clinicians, we can do diagnosis rapidly in the form of point of care testing. And even at home, you can now with the with the rapid evolution of technology, we can do diagnosis in our very home. So we have like this point of care test that we can do at home. And the most popular among these tests or among this platform is the glucose testing at home. So glucose testing is a form of diagnostic testing wherein you will know immediately if your glucose is high, low, or normal. So we have different type of testing or tests. In microbiological studies, we need to have a culture of the microorganisms that we are thinking the culprit of the infection that is associated to a certain patient. And later on, after we know who is the culprit or which bacteria or microbes infects the individual, we can do sensitivity. Another thing is the evolution of molecular diagnostics. Now, in our generation, we have a lot of molecular diagnosis or diagnostic tool to make a very reliable and accurate diagnosis of a disease, and especially infectious agents. Also, we have there the immunoassays. So first immunoassays was developed in 1960s, okay? And up to now, we have undergoing a lot of development to come up with a newer immunoassays that will help in the diagnosis of clinical infections and diseases, and also the imaging. So having said that, we need to have a sensor, okay? So for us to do all this testing, we need to have a sensor. What is a sensor? A sensor, guys, is a device that responds to a stimulus. If there is a stimulus, there will be a phenomenon that is happening inside or outside the body, which we might be sensing, okay? This sensor can detect heat, light, pressure, and also generate signals that can be measured and interpreted later on will aid us in the diagnosis of an infection or a disease. We have a sensor in our body which is natural to us, such as the nose. Nose is capable of sensing odor. So if we wanted to know the odor of something, our nose can detect it. So our nose serve as a sensor. Our tongue serve also as a sensor so that we can taste, okay? If this is sweet, salty, sour, so on and so forth. A sensor should be sensitive. This is selective to most natural compounds that we have. However, these are not selective to certain substances such as poison, other toxins, things like that. That's why there is a necessity for us to make a man-made sensor. So a man-made sensor is made up, or we call it as a biosensor. This biosensor, later on, I will be discussing to you what a biosensor is, but prior to that, let's discuss first the chemical sensors. Chemical sensors, this provides information about chemical composition of its environment. So anything about chemistry, the structure, is this a protein or a nucleic acid, so on and so forth, a chemical sensor can detect it. A chemical sensor consists of a transducer and a chemical recognition element. We need to have a chemical recognition element so that if there is this chemical present in whatever samples that we are dealing with, it will attach to this chemical recognition element, and then later on the transducer will recognize it, and then this transducer will make it digitalize so that there will be a result later on. Chemical recognition elements are selective, reversible, and has a detection limit. So having said what a transducer is the one that converts these physical or chemical changes into an electrical signal that gives us the result later on. So transducer could be instrument, any instrument that will detect binding, recognition of these chemical elements, so on and so forth. For example, a very good example of a chemical sensor is your pH meter. pH meter has the pH electrode, wherein we have a chemically selective glass membrane incorporated to the electrode of a pH meter. This recognizes the 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 generation of protons. So with that, how many protons is detected? The electrode gives numbers to that. And now, after knowing what a chemical sensor is, I told you before, let's discuss what a biosensor is. So a biosensor is a device integrating biological sensing element. Earlier, a chemical sensor integrates chemical sensing element. For a biosensor, we are talking about biological sensing element in intimate contact with an appropriate transducer. So we know that the transducer will will gather all the signal and later on will give us an output in a form of a result. So conversion of the recognition to a primary signal that can be amplified and subsequently processed. So the schematic of a basic biosensor is like this. So you have with you the analyte, could be whatever element is this, could be a protein, could be nucleic acid, could be a toxin, so on and so forth, and will attach to the receptor. Once attached to the receptor, this will be recognized by the transducer, and upon recognition of the transducer, this will create a signal that will be seen in a monitor or in a reading device that will give us the result. So for the history of a biosensor, as early as 1956, we are already having a sensor. Okay, so at 1956, we have already oxygen sensor, which was discovered by Clark. In 1962, an enzyme electrode, potentiometric enzyme electrode, spring instrument, bedside artificial pancreas in a bedside capability, needle type enzyme electrode for cutaneous implantation, so on and so forth. In 1998, there is a biosensor for organophosphate, which is a toxin. And also in 2000, we already have the glucose biosensor by many companies, which Medtronic's MiniMed implantable biosensor being the most popular in that time. And up to date, we have the point of care diagnostic test for COVID-19. So that is the evolutions. And as long as we are having issues with re-emerging infectious agent or infectious diseases, there will be a lot of biosensor that will be that we can create later on. So what are the application of this biosensor? So for home monitoring, we have the glucometer to detect glucose, especially for those diabetic patient, pre-diabetic patient. Pregnancy test, which is the most popular home monitoring test kit that make lateral flow assay a very, very interesting example of point of care test. And then we also have the COVID antigen testing. For the medical diagnostics, we have bacterial infection assays and HIV testing. Could be in the form of chemiluminescence or immunochromatographic assay in a form of lateral flow assay, so on and so forth. Also, ELISA, we have those for that, and the Western blot, of course, in HIV test. And we also have environmental monitoring for the pollutants in air and water. For the food industries, dissolved gases in the food, toxin in the food, and microbe or spoilage in meat or any meat products. And also application of this biosensor is very useful also in bioterrorism, especially when there is a biowarfare happening. We can immediately detect any virus or bacteria um, uh, that we are going to detect. So we have here COVID-19 tests. So COVID-19 has a lot of method for diagnosing this. Also, we have the immunochromatographic assay in the form of the lateral flow test strip and also the PCR. So PCR is a tedious and comprehensive laboratory test wherein you need to be a skilled person for you to do this, but thankfully we have already a PCR test done at home. And also thanks to the evolution of LFAs, we can detect the antigens or the COVID-19 antigen in just less than 20 minutes. So you have here the Binax, Lucira, Ellium, and a lot more. So in here, I am showing you a meat freshness sensor. So this is a form of a handheld biosensor which detects food spoilage. So especially we don't want to have a spoiled food for consumption. So we have a lot of biosensor that can detect that. So this specific biosensor or this device that I am showing you has the capability of detecting microbes or colony forming units in a meat product, and anything that reaches greater than 10 million CFU per grams is already considered as spoiled, and therefore we cannot use it for consumption, because if we do, we will be having a lot of infectious agents waiting to invade our system, and we don't like that. Also, we have this Minimed. This is also a very good biosensor. This is used by people who are diagnosed with type 1 diabetes mellitus. So what happened in this? This is an example of a in vivo sensor wherein this is incorporated to you that regulates the insulin in the body. So what happened in here? This device monitors the patient insulin level so that there will be no time where patient have a very low glucose level. If that is sensed by this device, the device will stop injecting insulin so that the body will regulate as a normal occurrence. So thanks for this small sensor that helps patients suffering from the DM without having checking from time to time manually if their insulin level is high or low, or maybe their glucose level is high or low. So that saves time and also saves lives, because if a patient suffering from DM and has a hypoglycemia already, that is a no-no, because it's in the middle of of a life and death situation, I may say. Also, we have, since the evolution of this technology, now we have a biosensor that we can just attach to our body in a form of stickers, tattoo-like sensors. So the one on your left is a wearable biosensor tattoos where which measures lactate levels in sweat. Okay, so this helps monitors the lactate level in the body, so that we will be monitoring if there will be like episode of cramps or any muscle anomalies. And this is very useful for marathoners. And on your right, we have this a patch that detects UV light, so exposure to UV light. So this is developed by L'Oreal. So this patch detects a level of UV ray exposure. So you just put it in your skin wherein there is a obvious exposure to sun or to UV light, and then this device will give you color as to the amount of UV that a person is exposed to. So measuring approximately 1 square inch in an area, and 50 micrometer thick, half of the thickness of an average hair strand. The patch contains photosensitive dyes that factor in a baseline skin tone, and change in color indicates exposure to UV light. So aside from this patch, some of these people or some of these companies already made other forms of UV exposure detectors. Some of them are in a form of a silicon band, and this silicon band, when exposed to UV, it will change color, and you will know, depending on the intensity of color change you see, is directly proportional to the amount of UV lights you are exposed to. And also, we have a peel on the camera. So this sensor helps in seeing, since there is a camera on a pill, this helps in seeing what is inside the human body, especially in the GI tract. So if you have like any obstruction in there, any suspect or something, if there's something wrong in the GI tract, this pill can help. And due to technology, aside from having a pill, having a camera in a pill, some of this pill already has the capability of dispersing drugs to the infection, to the site where you wanted to treat, or where you wanted to expose this to like to drugs. So we have those kind of technologies already, and this can be seen while this camera pill is inside the body. We have a device that we can see what's monitor what's inside, or what is being seen by this pill. So you will see if there's like something wrong from a device outside the body. And don't worry about this, because after the diagnostic procedure with this camera, camera on a pill, you just like excrete it excrete it out in a form of like maybe along with your stool. So no need for you to worry, because it will be excreted out in your stool. And this helps physician and healthcare provider for the diagnosis and rapid assessment of the status of the patient. So what are the advantages of a biosensor? Biosensor can detect low levels of analyte. Example of this, it can detect up to femtomolar or femtomole levels of a concentration of whatever analytes you are detecting. Also, biosensor provides an accurate results. Ease of use, do not require training for the users or for the end user. Will give you a fast, quick response when necessary, and provides online and continuous monitoring. And also, these are less expensive. So these are the ideal sensors. So ideal sensors should be accurate, selective, non-perturbing, reliable, low cost, and simple to operate. So those are the ideal sensor, and most of our sensors has these characteristics. So a schematic of a biosensor, as I have told you, whatever analyte you have in there, it will be recognized by a specific bioreceptor, as compared to like this picture below, wherein you have the analyte in as a red box, and then the receptor is a circle. Since they are not compatible, you will not be having any signal. But if you have if you have an analyte which is like this circle that will bind specifically or exactly to a bioreceptor. This will be identified by your transducer. The transducer will collect all the informations with regard to this. For example, how many of this, at what level, at what concentration. It will give signal in the form of numbers so that we have later on the results. So a component of a biosensor, biological recognition element, you have the enzymes, antibodies, DNA, etc. For the transducer, these are any devices that can recognize any contact with the analyte and a recognition element. And then later on, you have your computer or any device that will show you the results. So these are the different biological recognition elements. So we have enzymes, binding proteins, antibodies, nucleic acid, whole cells or cellular structure. Later on, for my sensor, I will be using a whole cell of a bacteria. This is a very interesting one because a bacteria is capable of detecting another bacteria. I will tell you why and how later. So transducer. So we have many forms of transducer. We have the one that is recognized if there's heat, if there is changes in electrical potential, if there is an electron redox reaction, if there is a light output, light absorbance, if there is a reactance or effects due to mass of the reactants or products. So these are the different types of transducer that we already have currently. So examples of platform for biosensors. So we can use a microtiter plate for us to detect this using a biosensor, lab on a CD, integrated sticks like your glucometer, paper strips like your LFAs, and then your lab on a chips for like microarrays. And now we have this technology. Thank goodness we have already an organ on a chip. So instead of us studying drugs in animal models or human participants, we can just use a organ on a chip for us to study the kinetics, the dynamics of drugs without invading or sacrificing animals, and also like exposing human to the adverse effect of these drugs that we are testing. So conclusion for biosensors are useful in diagnostics, environmental monitoring, food quality, and so on and so forth. Biosensors can be portable, home test kit type, and give us a high throughput. Biosensor can be cheap, simple, and easy to use. So those are the good thing about a biosensor. Okay, so having the discussion of what a biosensor is, let's now go to my project wherein I wanted to detect infectious organism infecting human in their urinary tract. So this is a rapid detection of urinary tract using a bacteria. If you are suffering from a urinary tract infection in your urinary tract, so when we say urinary tract, this involves kidney, urethra, bladder, and then bladder, urethra, and then there should be a culprit or a microbes that is producing the infection. So we wanted to test that as soon as possible so that proper stewardship of antibiotic treatment will be given to a patient. Sadly, because for us to really diagnose urinary tract infection, we have a lot of technology in diagnosing UTI. Especially culture and sensitivity is still the gold standard for UTI diagnosis. The problem with this one, with the urine culture, we need to wait for days, up to days or two, or it can be prolonged to maybe a week before you can get a final diagnosis of UTI. So with this, we develop in our lab, especially myself, I develop a way for us to detect bacterial load in the urine with the use of another bacteria. So in here, we are using a whole cell biosensor in the form of a luminous bacteria. So this luminous bacteria is ubiquitous in nature. We can easily isolate this anywhere. And in the US or anywhere, we have occurrence wherein bioluminescence show or bioluminescence sightings is found in some oceans in the world. So we can see glowing oceans in Malta, wherein there's like an ocean called the Blue Grotto. So bioluminescence is seen in there. Bioluminescence is seen in the Bay of Puerto Rico. In San Diego, California, you can also see bioluminescence sightings in there. Here in Florida, where I am at now, we can see also sightings of bioluminescent in an ocean, in Japan, and also in Taiwan. But for my specific bacteria, I have isolated this myself from a squid. So I isolated the bacteria from a squid, purified, and later on for the sensing of my my urinary tract infection, or with for the detection of the bacteria in the urine. So what are bioluminescent bacteria? So bioluminescent bacteria or bacterial bioluminescence is a luminescence or a light produced by a bacteria. The emission of cold blue light from the metabolic activity. The good thing about bioluminescence is that it doesn't need any excitation. No excitation is needed as compared to fluorescence, where you need to excite something so that you will have the light production. For bioluminescence, no need for you to excite anything. In its natural state, this bioluminescent bacteria will just produce light, especially when they sense a quorum within them. There is this thing in the field of microbiology wherein we said that these microbes is capable of talking to each other or communicating with each other, and this is in a form of quorum sensing. Quorum sensing is the method or the way this microorganism communicate with each other. Also, in disease progression of a bacterial pathogen, quorum sensing is essential also. For example, in Pseudomonas also provides quorum sensing, wherein when they do release the quorum sensing molecule, they tend to make themselves more virulent or more pathogenic. But in this case, this bacteria, as long as there are many in a population, they produce light. And that is how bacterial bioluminescence is. So there's a lot of marine luminous bacteria. We have Photobacterium lugnati, Photobacterium phosphorium, Photobacterium mandapamensis, Vibrio fischeri, Vibrio harveyi, Vibrio splendidus, Logae orientalis, and even Alternomonas hanidae is a bioluminescent bacteria. But for my thing, I use Photobacterium lugnati and Photobacterium mandapamensis as my whole cell biosensors. So this can be isolated easily. So what you can do, collect water, seawater, or swab sample from fish or squid, and then put it in a luminous medium, and then incubate it at room temperature. And when it grows, go to a dark room. If there's like something that glows, isolate it, purify it, and now you have your own bioluminescent bacteria. And you can do whatever you want in there. So if you wanted to court somebody and impress this person, you can say, or you can tell your feeling in a form of writings using this bioluminescent bacteria in a plate. Put there that you this person is special to you. So you can do that. Whatever you wanted, you can do that. So how this occurred? So this is because of a luciferin and luciferase interaction. So luciferin-luciferase interaction, this makes this thing glows. So what is a luciferin? Luciferin is a pigment in an organism with a bioluminescence, which is composed of chemicals, sulfur, nitrogen, and this luciferin binds to an enzyme. And when this binds to an enzyme, this produces light. And this enzyme is your luciferase. Luciferase is an enzyme, a protein that is associated with luciferin that produce light. So easy. You have luciferin, couple it with luciferase, attach, glow. So that's how light is how light is produced. Unlike from fluorescent, you need to have excitation. Wherein in here, no, you just need luciferin, luciferase, bind, poop. You have your light. So there, this light is cold. So this is a cold light. So blue light are cold light. So therefore, there is no energy wasted as heat. And this light is at 490 nanometer wavelength. So this is blue light, cold light. So these are other forms of luminescence wherein you can find in salamanders, fungi. You can also find it in crawfishes. Also, you can find it in nematodes. There's a lot of bioluminescent in the world. But for my thing, I use a marine source for my bioluminescence because this marine source provides a very good signal of bioluminescence. So the wavelength of your bioluminescence is at lambda maximum of 490 nanometer, which give you a blue-green light. The light is cold, as I have told you, since the conversion of chemical energy to radiant energy is very high. So there is no energy wasted as heat. And this luminosity or the brightness is tied proportional to the well-being of cells. This luminescence can be affected by pH, salinity, dissolved oxygen, temperature. So if the cells or if this bacteria are healthy, therefore you will be having a bright bioluminescence. And if the cells are unhealthy, and therefore you will observe a darker background or a dimmer bioluminescence. And this dimming and darkening of bioluminescence or extinction of bioluminescence is affected of pH, salinity, dissolved oxygen, temperature. And therefore, you can use this as a sensor later on for any analyte that you wanted to observe. There's a lot of researches that uses bioluminescence in their field because bioluminescence can be easily acquired, can be easily modified, can be easily grown in culture in the lab. You just need to like transfect it to E. coli. So your E. coli that is just E. coli, you can add a plasmid in there, modify, and then later on you make a bioluminescent E. coli, wherein later on this bioluminescent E. coli, you will ask this E. coli to produce multiply, so that later on you will be getting the protein that you wanted for luminescence. So you can do that easily. And now, since we know the good thing about the bioluminescence, so let's now discuss what a urinary tract infection is. So urinary tract infection is an infection of any part of the urinary system, from the urethra, bladder, ureter, kidney, and then UTI or urinary tract infection is the most common infection, bacterial infection, affecting 150 million people worldwide each year. And then preventable hospitalization is possible if this urinary tract infection is assessed and diagnosed correctly in an early stage. And sad to say that 1 in 2 women will suffer UTI within their lifetime. Urinary tract infection, guys, is more common in female than in men due to the anatomical position of their urethra from the anal area. So since the proximity of the urethra to the anal area is discussed in here because anal area is where your E. coli lodge. It is common for you to have E. coli in that area, but not in the urethra. So what happened is, if the female is very unhygienic, this microorganism from the anal area will transmigrate to the urethra and then later on cause infection. So how that happened, I think I'll be discussing that to you later on in the next couple of slides. So urinary tract infections, this is common. The most common pathogen associated to UTI is E. coli. Okay, so uropathogenic E. coli makes most or 75% of all the UTIs diagnosed worldwide. But there are other microorganisms such as Klebsiella pneumoniae or Klebsiella pneumoniae. If this is associated to urinary tract infection, you need to take consideration if there is renal calculi, because renal calculi is associated to some of these pathogens such as Proteus mirabilis and sometimes Klebsiella, and urea production is associated to this, since Klebsiella pneumoniae is a urea loving organism. So your urine has a lot of urea. That's why Klebsiella is second most common to this. This is so your urine is giving Klebsiella a good place to survive because of the urea found in your urine. Later on, you have Pseudomonas for the gram-positive microbes. We have Staphylococcus saprophyticus. This Staphylococcus saprophyticus is the pathogen of concern to patients who are sexually active and patients who often use tampons. So if you have your UTI associated to saprophyticus, it could be because of tampon use or sexual activity. But still, aside from that, there are other microorganisms that can cause urinary tract infections, such as your Enterococcus species, Streptococcus agalactiae, and Staphylococcus aureus. The Staphylococcus aureus is also a culprit because Staph aureus is found abundantly in our skin. So Staph aureus is okay to be seen on the skin, but when this Staphylococcus aureus goes inside skin due to like wound infection or vertical contamination, then this can cause UTI. So there. And this is the pathophysiology of urinary tract infection. First, it will contaminate the periurethral area. And then later on, when there are like massive growth of this, there will be colonization. After colonization, this microbe tends to go up into the bladder, leading to cystitis or urinary tract infection. And in the urethra, there will be like invasion, intracellular replication, flux and filamentation, and then later on it will migrate up to the kidney, wherein you will be having pyelonephritis. And later on, since the kidney has a vessel in there, we have like the afferent interior arterioles. This is where this E. coli will go to the bloodstream, has the capability of going to the bloodstream, and if this reaches the bloodstream, multiply in the blood, it will now lead to bacteremia. That's why urinary tract infections should not be take for granted, because these can lead to a more severe infection, which involves bacteremia, and severe bacteremia will lead to sepsis, and then later on, death will occur. There are many methods of UTI diagnosis. So we have urinalysis, wherein we do physical, chemical, and microscopic analysis of the urine sediments. If there will be like bacteria found in the urine, you can use that. Also, a strip test known as the leukocyte and the nitrite testing. This is rapid, but the problem with this one is that the specificity and the sensitivity of this varies. The predictive values is very has a reputation of poor to good percentage. And also the gold standard is the urine culture and sensitivity. The lower limit of a positive UTI diagnosis is 100,000 CFU per ml in a normal infected patient. And this takes 2 to 3 days for a final diagnosis because you need to have the organism grow on a medium, on a synthetic medium outside the body done in the lab. And then after that, we do antimicrobial susceptibility testing for us to know which better antibiotic can be given to a patient to kill or destroy this invading pathogen. So molecular-based diagnosis versus traditional. So we have also molecular testing for UTI, but the problem with this one, yeah, this is rapid, less than 24 hours, you can have a result, but this could be expensive. While the traditional culture could be outdated, results is outdated because the technology is from 1928 up to the present, and there's no much how you call that? There's no much evolution on how you do cultures. But now, thanks to the evolution of technology, we have other methods aside from culture that will give us a good monitoring capability or diagnostic capability in UTI, but it will be either expensive or tedious to perform. So this is a table wherein we can find the different technology for UTI diagnosis. Some of them are point of care, some of them are rapid and inexpensive, but the technology that I have developed will only take 5 minutes for you to say if there is a urinary tract infection or not with a patient. With the consideration of how much bacterial load is present in a patient urine. So aside from UTI, the problem is antimicrobial resistance. So we have a lot of antibiotic resistant strain of microbes in the world today. And this is the premise. The more antibiotic we use, the more resistant we will be observing. This is because of many factors. So each year, antibiotic-resistant bacteria and also fungi cause at least an estimated 2 million infections in the world that contributes to 35,000 deaths in a year. Plus, there is also the progression of or any related antibiotic-related infections, such as the infection caused by Clostridium difficile or Clostridioides difficile. This microorganism, we call it as the silent bystander because this organism attack when there is no other organism is present in the surrounding, which are affected by antibiotic treatment. So those normal flora have died already due to antibiotic treatment. This Clostridium difficile can resist this antibiotic. So when they resist antibiotic, they grow rapidly and then infect individual. And this contributed to 223,000 cases in the world and gives 12,000 deaths per year. So antibiotic resistance happens when there is overprescription of antibiotic, patient not finishing treatments, antibiotic treatments, overuse of antibiotic in livestock. So if companies use antibiotic in the livestock or in poultry, in animal husbandry, and then this product such as like meat is dispersed or dispensed to human consumption, the remnants of antibiotic presence in the meat product can also affect and contribute to resistance with antibiotic. So among the 18 alarming antibiotic resistance threat, we have 6 alarming and common resistance, which affects more than 4.6 billion US dollar annually. So we have here enterococcus resistant, vancomycin resistant, enterococci, carbapenem resistant, acinetobacter, methicillin resistant or the MRSA, CRE carbapenem resistant, enterobacteriaceae, the ESBL, extended spectrum beta lactamases, and the MDR, the multi-drug resistant strains. It is said that in 2050, there will be more and more and more deaths due to AMR. So we wanted to prevent that when 2050 came. So how does microorganism do resistance? A lot of microorganisms do resistance due to their plasmid. And also some of them can modify the target and also activate the pumping out mechanism. So when there's the antibiotic in there, this microbe has the capability of flushing away this antibiotics, and therefore the antibiotic will not be effective. So those are the different mechanisms of antimicrobial resistance. Okay, so having said that, we wanted to develop a test which can simultaneously detect UTI bacterial load quantitatively and detect the presence of antibiotic-resistant bacteria. And that will address a major gap in a point-of-care diagnosis of UTI in a manner that has not been demonstrated previously. So this is the concept. So UTI detection, you have your urine, it has the bacteria in there, E. coli. You add bioluminescent bacteria. This bioluminescent bacteria is very sensitive to many aspects, to many considerations. So it could be presence of the bacteria itself can be affected or can affect the light production of this bioluminescent bacteria. So without the presence of this E. coli, just the bioluminescent bacteria, it will give you a bright bioluminescence. But when there is an interaction with these microbes to another microbe, which is E. coli, there is a depletion or decrease in light intensity. That is how we foresee before how are we going to detect UTI. And now we wanted also to incorporate AMR detection. So what we are thinking is we have a cephalosporin. This cephalosporin is caged with colanthracene. Colanthracene is a substrate that can give bioluminescent on a protein GLAC. So when colanthracene binds to GLAC, it will produce light. So how this happened? So cage cephalosporin. So this is your cephalosporin and this is your colanthracene. We bind them together, and for us to release this antibiotic, we need to have an enzyme which is a lactamase, okay, which will be present to a bacteria found in the urine. Since this bacteria in the urine is producing lactamases, the lactamases will be cleaved outside and can attach to the colanthracene. Since cephalosporin is a beta-lactam drug, these beta-lactamases can destroy the binding between colanthracene and cephalosporin, releasing the colanthracene and cephalosporin. And once the colanthracene is released upon addition of a luminescent protein, which is a Gaussian luciferase, it will bind to the colanthracene and we produce light. So if there is AMR detection due to the presence of beta-lactamase that cleaves these two, cephalosporin and colanthracene, we produce light. So that is our thinking on how we are going to detect AMR. So before we proceed to like the point of care platform for UTI diagnosis, we first do it in a huge tube technology. So I call this tube technology as the tubetior or the tube bioluminescent extinction technology. This is simple. You have the urine, add bioluminescent bacteria, mix, and then after mixing, wait for decrease in bioluminescence. If there's a decrease in bioluminescence, there is the presence of another bacteria in the urine, and you can detect UTI rapidly. And with that, we test it with 4 possible cause or possible uropathogen: E. coli, Staphylococcus aureus, Proteus mirabilis, and also a fungi, which is Candida albicans. And you can see in here in this graph that using or detecting this microorganism in a urine, we see that as the number of cell increases, the highest the decrease, or the more decrease in bioluminescence we observe. So in here we have like million CFU per ml. If we have like more cells, there's like an immediate decrease in bioluminescence. With this technology can detect any microbe at that high concentration, up to 100,000 CFU per ml. But this technology cannot identify specifically which pathogen caused the infection. But the good thing with this, as long as there's a uropathogen present in the urine at a high concentration or up to 100,000 CFU per ml, we can say that the patient has UTI. And now we know that this can be done in a tube technology. We wanted to put it in a micro scale. So we put it in a micro titer plate. So instead of a test tube, we just like minimize the or use a small volume of the urine and also the bioluminescent bacteria, and then test it using a highly sensitive luminometer. And with that, we also have the same findings as compared to the tube extinction technology. As the number of cell increases, the more decrease in signal intensity is found as compared to just negative control, which give us a very, very high signal intensity of luminescence. And this give us a linearity of about 99% linearity. Since we know that this worked on a micro scale, now we wanted this to be usable in a settings wherein there's no need for us to worry with the stability, with the containment of the bacteria, how long can we use the bacteria. So we tried lyophilizing this bioluminescent bacteria. So we freeze dried this bioluminescent bacteria, and we check the bioluminescence of this microorganism up to 3 months, and we observe that at room temperature, this signal intensity is not affected at all. So we can now say that, oh, this can provide us a better way of preserving this bioluminescent bacteria, so that later on, when we wanted to use this, the number or the signal or the intensity of light this microorganism is giving us are still the same. I am only showing up to 3rd month in here, but after the 3rd month, we still do prolonged incubation or prolonged storage of this lyophilized bioluminescent bacteria, and we can still observe the same signal intensity of up to 8 to 9 months. So that is how very good we can preserve this microorganism using a specific specific protectant for the bioluminescence to be stable at that longer period of time. So what we do after we lyophilize this, we try this lyophilized bacteria to be used in the same technology as what we did in the microtiter plate. And at 4 consecutive days, we have the same consistent results. And we know that this consistent result will give us a very good platform in making a point-of-care test device or point-of-care platform. So why this work? We know that this worked, but we also wanted to know how this thing worked. So we considered 3 different things. Is this because of quorum sensing? Is this because of the toxin production of the microorganism? The E. coli produces an invasive or whatever kind of toxin that interfere with the bioluminescent, or is it the bio-oxygen demands? So what we did, or what we do, we try using a specific strain of E. coli, which is an NEB5 alpha cells or DH5A, DH5 alpha cells, which is an E. coli that is chemically devoid of everything. So it's just like E. coli without anything. So this E. coli doesn't have any quorum sensing molecule, doesn't have a toxin. It's just like a plain E. coli, just there to grow. Okay, so we tried using this. So the good thing is that when we use this chemically modified E. coli, we observe that even at a very at a high concentration, we still observe decrease in signal. So we thought of, or we think of, or we concluded that, oh, this is not because of quorum sensing. This is not because of toxin production. Because why? Because this E. coli, specific E. coli, is devoid of quorum sensing molecule, devoid of toxin, devoid of anything. If this E. coli has those quorum sensing molecule and toxin, the level of the light intensity should all be either high or low. But in this case, we have a gradient of signal intensity. Okay, so we know that this is not because of quorum sensing, not because of toxin production. Now we have the bio-oxygen demand. So for us to rule out if this is because of bio-oxygen demand or whatnot, we try to do bio-oxygen demand assay. So we tried removing all the oxygen presence in an artificial urine and in the ionized water. So how we do that? So we purge this artificial urine and the ionized water with argon. So we replace the oxygen with another gas, which is argon. Okay. So and then later on tested these samples, or the ionized water or the artificial urine. So for the ionized water, we have oxygenated, which is not purged with argon, and then the oxygenated one is the one purged with argon. And we see that at an oxygenated ones, we observe that there is a high signal intensity as compared to the purge, the ionized water. Similar thing in an artificial urine. So what happened is like this. The E. coli found, or any pathogen found in the urine, is a highly aerobic pathogens. This pathogen needs oxygen to grow. So what they do, they consume all the dissolved oxygen or any oxygen present in the matrix, okay, for them to grow, and then leaving the matrix deoxygenated. The bioluminescent bacteria is a highly oxygenated or highly aerobic organism, wherein since the matrix is already devoid of oxygen, or there is a depletion in oxygen due to the utilization of the uropathogen, upon addition of this microorganism, since they need oxygen, for survival or for the production of light, they will not produce any light. That's why we have a low light intensity in a deoxygenated one and a high light intensity in an oxygenated one. And that concludes why we are having the positive and the negative results in the UTI. So now we know what is the cause of the extinction. It is because of the bio-oxygen demand. So we have a lot of questions. Maybe if there is another substance or whatever chemical present in the urine, it can interfere. So we try to do interference study. We try adding glucose, changing the pH, and adding urea in the urine, and we find no signal intensity, and there is a no significant difference in the light intensity produced of the negative, which is just the urine, and those spike with glucose, pH, and urea. And now we know that there is no interferences, whatever is happening in there. So this is just because of the presence of the microbe in the urine. That's why we are getting a decrease in signal intensity, and therefore we can conclude that we can detect UTI using that principle. Now we know that we can detect it. We wanted now to detect antibiotic resistance. Is this microbe present in the urine has a capability of having an antimicrobial resistant genes in there. So technically, we have the urine. We expose the urine with an antibiotic, and then later on mix, add luciferase. And what happens is that if there is antimicrobial resistance, there will be light production. And if there's no antibiotic resistance, there will be no light production. This is because of the exposure to antibiotic in the urine. And we do that, and we have tested different E. coli that has a resistant gene. E. coli resistant gene. We have E. coli with ampicillin resistant, E. coli with chloramphenicol resistant, E. coli with trimethoprim resistant, and E. coli with fluoroquinolone resistant. So we have this so that each mechanism of action, we have a representative of each mechanism of action so that we can cover a huge AMR spectrum. So ampicillin is for beta-lactam drugs, chloramphenicol for protein synthesis inhibitors, trimethoprim for folic acid synthesis inhibition, and fluoroquinolones for DNA synthesis inhibition. So we cover a huge range of mechanism, antibiotic mechanism of action. So you can see in here that just the E. coli without any resistance antibiotic, we observe that there is a high signal intensity as compared to those with antimicrobial resistance. We observe a decreased signal intensity. So in here, when there is an antibiotic resistant found, there is a decreased signal intensity. So we tried also doing an test wherein we have the ampicillin resistant E. coli, and this ampicillin resistant E. coli is exposed to ampicillin, chloramphenicol, trimethoprim, sulfamethoxazole, and fluoroquinolone. And you can see in here that when an ampicillin is exposed to ampicillin, since it is resistant to that, we observe that there is a growth in the organism. Oh, by the way, guys, in here we are detecting the ATP levels of this microorganism. So when they are growing, when the bacteria is growing, we observe there will be a lot of ATP production in the matrix. So this ampicillin-resistant strain of E. coli is exposed to ampicillin as expected. Since it is resistant to ampicillin, it will grow in an environment where ampicillin is. And good thing, or good enough, we have like a high signal intensity as compared to exposing this to chloramphenicol, trimethoprim, and fluoroquinolones, wherein there is a decreased signal intensity. And then later on, we tried this in a multi-drug resistant E. coli. And we exposed this to these antibiotics, ampicillin, trimethoprim, and fluoroquinolone, and still we are detecting high signal intensity, and there is no significant difference between the results of the positive control up to those with the MDR resistant strain exposed to ampicillin. So we are expecting, since this is MDR, when we add ampicillin, this is resistant to that, it grows. Same with trimethoprim and fluoroquinolone. So this is just a summary of all the figures that we have in here. Okay, now this is the proposed POC system. So we wanted this to put in a scale wherein easily accessible to the end user. So with the use of just a cell phone, we can detect UTI as well as antimicrobial resistance. So this is the adapter, that is the cartridge. You add the urine sample and the bacteria, and then the bioluminescent bacteria, and then the luciferase. And in here, mix. So if the patient is having UTI, so you see in here, we have plenty of wells. These first 3 wells in here is for the detection of UTI. So positive UTI is no bioluminescence. Negative is with bioluminescence. And for the antimicrobial resistance, positive is no bioluminescence, and the negative is with bioluminescence. So for example, the urine sample is positive for a pathogen, is having microorganism in the urine. We have blackout or darkening of or there's no bioluminescence. And if there is antimicrobial resistance, it will give you a decrease in signal intensity. And then by taking a picture, it will be analyzed by a program in the cell phone, and then later on wait for like certain time, it will give you results such as UTI positive, AMR, fluoroquinolone resistant, kind of thing. So this is the prototype. So we try using the results gathered by the cell phone cameras, cell phone picture, and then by a luminometer, which is a highly sensitive luminometer. And we found out that there is no significant difference between the reading of these two platforms, like the ImageJ cell phone picture, and then the results using a highly sensitive luminometer. So now we know that we have that. So we wanted also to make it easy. What if the person doesn't have a cell phone? We try it on a point of care UTI technology by using a handheld luminometer. So with this project, we try using a handheld luminometer, and then we test this for the AMR. So we do handheld luminometer reading versus the highly sensitive luminometer. So ClearUStar is a tabletop luminometer which is highly sensitive. We do the testing using this luminometer, and we use this method that we incorporate using a handheld luminometer to a desktop luminometer. And here are the results. So we are detecting the ATP levels because that is an ATP based assay. The handheld luminometer is based on the ATP level to the detection of bioluminescence. So with that, using a ClearUStar, so wild type E. coli has a lot of ATP because we expect that this grow, okay, and this E. coli exposed to ampicillin, chloramphenicol, ciprofloxacin, the signal intensity or the ATP will be decreased because they are exposed to antimicrobial drugs. So therefore, there will be like a decrease in ATP, which is true enough that there is a decrease in ATP observed in ampicillin, chloramphenicol, ciprofloxacin, and trimethoprim. This is using a highly sensitive luminometer. Also, we wanted to test with ampicillin-resistant E. coli exposed to various antibiotic as what we have done before, and we expect that in wild type and AMR-resistant E. coli exposed to ampicillin, it will be growing. So therefore, there will be high ATP level. And comparing this ampicillin-resistant E. coli exposed to chloramphenicol, ciprofloxacin, and trimethoprim, there will be like a decrease in ATP level because they are suppressed by these antimicrobial drugs. Same thing with chloramphenicol resistance. You observe that there is a high ATP level in wild type and in chloramphenicol resistant strain, but in a chloramphenicol exposed E. coli, there's a high ATP, and for the ampicillin chloramphenicol-resistant E. coli exposed with ampicillin, ciprofloxacin, and trimethoprim, there is a decrease in ATP levels because they are suppressed or they are inhibited by this antibiotic. So that's good. And we compare it with the handheld luminometer, and here are the results. So for the wild type, the ATP level is high as compared to those exposed in various antibiotic, ampicillin, chloramphenicol, ciprofloxacin, and trimethoprim. We observe that there will be a decrease in ATP level, which we have in here. Same thing with ampicillin-resistant E. coli, ampicillin-resistant E. coli exposed to ampicillin, and then just the wild type, we observe there is a growth. And we see in here, as compared to ampicillin-resistant exposed to chloramphenicol, ciprofloxacin, and trimethoprim, there is a decreased ATP level on these samples. ATP levels of chloramphenicol, same thing. Chloramphenicol resistant E. coli exposed with chloramphenicol, we expect there will be high ATP level with the wild type also, but with ampicillin, ciprofloxacin, and trimethoprim, we observe a decrease in signal, ATP signal. And then we tried comparing the results of Clarius Star and this handheld luminometer, and we found out that there is no significant difference between the reading of the two. So using Hygiena, we can immediately detect, like Hygiena is the device, brand name device, as compared to a highly sensitive one. So we can effectively use this handheld luminometer in detecting antimicrobial resistance using an ATP-based bioluminescent assay. And with that, I would like to thank my mentor, Dr. Sapnadeo, Dr. Sylvia Donner, Dr. Yizu Fengong, Dr. Zing, Dr. Dikichi, and all of the people in our lab. So we have a diverse people in here. So each people that you see there is working on a different biosensor project or any drug delivery, any microbiome studies and cancer researches. Also, I would like to thank Fulbright for the opportunity that they gave me the opportunity of doing a research and studying here in the United States, for the funding, NIH, NSF, my university in the Philippines, University of Santo Tomas, and the Far Eastern University Nicanor Reyes Medical Foundation. And with that, I am willing to take any questions from the floor. Thank you for listening. Hope you understand my presentation. Thank you, guys.
Are there any questions from the attendees? Just check. Nothing on the Q&A.
That was really wonderful. Thank you, Shermin.
Thank you.
Very
Thank you, ma'am.
informative. Point of care. Wow.
Yeah, thank you. I also made like an ELISA, but we can't call it as an ELISA because we didn't use an enzyme, but therefore a bioluminescence in detecting the spike antigen of COVID-19 using a fusion protein created by our lab. So our lab created a bioluminescent fusion protein to attach and detect the spike antigen of COVID-19, and successfully we are able to detect that.
Dr. Ring, we have
Thank you, Sir Erwin.
same question from SDS. Impressive. How long did it take you to do this?
So I start the tube bioluminescence assay. I have the preliminary study on that in 2014, and I have like the opportunity to develop more on that. I started developing it in 2018 when I started doing my research here in the United States, and that only takes me 2 years for me to like get those results. For the UTI technology, and for the AMR, I started the AMR in 2021, and in 2022 I am already getting, like last year, I'm already getting good results with that.
Okay, there's one more from Valerie. That was incredible. I'm wondering how soon this will be implemented for general use.
So my mentor and I were still like I don't know the answer, but once we already have all the documentation, things like that, I think it will be available anytime soon. And also the target for this one, that's why I developed this, is that I came from a how you call that, like a developing country wherein the access to highly or sophisticated, in some part of the Philippines, wherein the access for a highly sophisticated method for diagnostic is very rare. So we wanted this to reach this low income countries, so that they can have like a reliable and a rapid way for the detection of UTI. Because even at the hospital settings or in the field, you can just like get a sample, mix, take a picture, you can have a result.
Oh, wow. Okay, from anonymous attendee, this is a great, thank you. Another anonymous attendee, great presentation. I had some trouble with the video. He has such a great passion. I hope he gets his method distributed. Is this for the public or high complexity?
This is for the public. As what I have told you guys, this is like the main purpose why I developed this is to reach on a low to those low income countries that do not have an access to highly sophisticated method in diagnosing infectious diseases. Maybe later on, after the UTI, we can verge on other infectious diseases or whatever disease we have, and then use the same technology. Hopefully we can do that.
Okay. Here's an interesting question. What made the projected cost for handheld camera detection methodology? Cost benefit with home testing.
Um, when we are, since everyone has a phone, this will be like the cost of this, as we computed before, this will be each test will be cost less than $1,
Oh, wow.
because you
Okay.
don't need to buy the reagent, because the reagent is free. I just isolated it from the marine source, lyophilize it, and lyophilization will not take you a lot of money, and it's really like inexpensive.
We have another question. I have heard UTI causes confusion in older adulted. Is this true?
What you mean, like the occurrence of repeat like recurrent of UTI in adult in adult population? Is that oh yeah.
Yeah.
So this one, since we wanted to like uh uh touch on the recurrence of UTI, we still wanted to have a wide range of trials with repeated or recurrent UTI in adult, and I think this will give us like a very positive very positive uh very positive result in detecting recurrence in UTI.
Nice. Okay, we have one more. You are profit-led in your developing this concept and translation to diagnostics. Comment.
What you mean by profit led? Like we can sell it?
You are profit.
So like this project is funded by funding agencies, so I don't know. Oh, I see.
Okay. Anonymous attendee said, I mean, you are altruistic. Okay, let's go to Anne Gregorio. Wow, that will be great to use not only in the hospital, but also in reference lab. Great presentation.
Thank you, guys.
Okay, any more questions? Oh, let me look at the. Um, all right, I think there are no questions. Oh, wait, we have one attendee who raised. Oh, Valerie. Go ahead, Valerie.
I just want to say, as the president of Chemalt and one of the coalition members, I am so impressed with what you're doing, and just it's such an important thing to bring low cost, quick tests of this nature to underserved populations. So I wish you well with your luck, and thank you for making time to speak with us. I'm very, very impressed, and I appreciate it. Thank you so much.
Thank you, Ma'am Valerie. Thank you.
Okay, are there any further questions and comments before I give out the page
Yeah,
number?
that's why we wanted to have like a specific or rapid diagnosis of UTI, because we don't want this to go into sepsis later on. Because when sepsis, it's already like difficult to to revert and manage. So as early as possible, we wanted to detect UTI. Will there be resistance to antibiotic? If there is, and then there is a good stewardship of antibiotic later on. So yeah, hopefully it helps.
There's another comment from an anonymous attendee. Many elderly people develop a URI to urosepsis, or do you mean UTI equals quick diagnosis, TX?
Yeah, I think that is what I I said before. Like that's why we wanted to diagnose it rapidly. Yeah. So the good thing about this is like two bacteria is like detecting each other.
Hmm.
Thank you guys.
Thank
Appreciate it.
you, good job. Thank you.
Thank you, Sherwin. That's a great presentation. Thank you so much.
Thank you, ma'am. Thank you for inviting me.
Okay, the page number for this session is 8105. Gonna type it in the chat box.
Did I made it on time?
Yeah, it's close. It's close to 540. Yeah, it's 538. Sorry, let me type it in. Sorry. 8105. I have one more question. This is last. When you were explaining, are there any limitations for this bioluminescence method? Are there like limitations when performing this kind of
I think the limitation that we have in here, since the organism that we use for the model of organism that we use for this project are all aerobic. Maybe if there is an anaerobic microbes that cause infection in the urine, then I don't know if we can detect it.
I see. Interesting. Very interesting. Okay, I want to ask again our attendees, any questions, comments?
It says the number again.
The number again. Okay, that's 8105. Base number 8105. Check out the webinar chat. I typed it in. We made it on time. It's 5:40. Thank you, Dr.
I'm so
Sherman.
scared because I thought I will be using a lot of time, because sometimes when I tend to talk, I talk too much, without noticing the time. But good thing, like I look at my watch and like, oh wait, I need to slow down.
Nice, nice, nice. Okay. All right. Thank you, Dr. Sherwin. We have a Saturday night social. So anyone interested to log in, please do so at 6 p.m.
Yeah, thank you again, Sherwin. And that's right, Apollo. We would like to invite everyone for to join us for some social nights. Probably we can have some wine, cheese, or coffee. It's cold. Join us,
heavy
and
coffee.
it's a good time to get to know more about the coalition, or just relax and talk what's happening out there. We are here in La Crescenta, and we are experiencing snow. So everybody's outside. It's really, really nice. I don't know. We're like kids running outside, running under
don't
the
feel
snow.
it.
But yeah,
Mm.
so thank you again, Sherwin. And we again would like to see you in a bit on our next session. Please come and join us.
Thank you, Ma'am Boots. Thank you, Apollo. Thank you, everyone. Thank you, Ma'am Emma.
Night.
Good
See you guys on social night.
Can I leave na?
Thank you.